So if we had the ability to tell groups of cells what to build, we would be done with birth defects, with traumatic injury, meaning we'd have regeneration, we could reprogram cancer, aging, degenerative disease. All of these things would go away if we could communicate our goals to a set of cells. And so this anatomical compiler fundamentally is not a 3D printer. It's not about putting micromanaging the positions of cells where you want them. It's about communicating. It's a translator, really. It's about communicating your anatomical goals to the mechanisms guiding the set points of the cellular morphogenesis. Now, why don't we have this? And we have nowhere near that kind of capability. Now, why not? Molecular biology and genetics have been going gangbusters for decades. Why don't we have it? I'll give you a very simple example. Here's a baby axolotl. It's a salamander. The babies have little legs. Here's a tadpole of a frog, Xenopus laevis. They do not have legs in this stage. In my lab, we make a chimeric construct called a frogolotl. So we take a bunch of cells from Xenopus and a bunch of cells from axolotl, and we make a frogolotl. Now, I ask a simple question. We have the genome of the axolotl. We have the genome of the frog. We have complete genetic sequence for both of these animals. Can anybody tell me whether the frogolotl is going to have legs or not? And the answer is no. We have absolutely no formalism, no models that will make a prediction on this. And that's because this kind of thing is not a decision made at the level of molecules or genes, and it's not made at the level of individual cells. It is a collective decision, and we still do not have a good understanding of how cellular collectives make decisions. We're very good at manipulating information like this, which molecules talk to which other molecules, but we're a long way away from control of large scale form and function. And the reason I think is because molecular medicine is still stuck where information technology was in the 40s and 50s. This is how you programmed a computer in the 40s and 50s. You physically had to rewire the machine. You were down at the hardware level. And this is where modern biology is. All the excitement is around DNA editing, pathway rewiring, protein engineering. All of these things are directly, single molecule approaches. All of these things are directly at the hardware level. And what we're still leaving on the table is really the software of life, specifically the competencies, the intelligence, and the problem-solving capacity of the material, which is very different than all of the passive matter that we've been engineering with for millennia. So when I talk about intelligence, I do not mean a human level metacognitive ability to know that you're smart and then to know what your goals are and so on. I mean something much more basic and fundamental. This is William James's definition. It's the ability to reach the same goal by different means. So what we have here is a continuum, and James actually talks about this, the continuum between two magnets trying to get together and Romeo and Juliet trying to get together. The difference being that these magnets, if separated by a barrier, are never going to go around and meet each other because they don't have the ability for delayed gratification. They cannot go further away from their goal in order to later do better. Romeo and Juliet have all kinds of ability of planning and so on. And in between, you have lots of different systems, cells and animals and autonomous vehicles and robotics and all kinds of stuff that has different degrees of competency to reach their goal when confronted by a barrier. So the idea is that you have to force them to try to use different means. You can't do this from observational data. You have to do perturbative experiments to put some kind of a problem between them and their goal, and then you have to see what level of competency they can muster. So, okay, so what kind of collective intelligence do we see in cellular swarms? Well, first of all, so development is quite robust, it's quite reliable, and that's great. Most of the time, a normal early embryo will become a normal, it will have a normal human morphology. But the amazing thing is that actually it's not hardwired, because you can cut early embryos into pieces. You can cut them into halves, quarters, eighths, and so on, and you don't get a half body. Each piece will give rise to perfectly normal monozygotic twins. And so if this is anatomical morphospace boiled down to two axes, you can have this ensemble of states associated with a normal human target morphology, this goal state here, and you can get there from fairly diverse starting positions, maybe avoiding some local minima along the way, but you still get there. So that's interesting. Development, regulative development, is able to get to the same goal by different paths. Okay, so different means to the same goal. This is not just for embryos. So some animals like the salamander can do it throughout its lifespan. So amputate the arm anywhere along. In fact, they also regenerate their eyes, their jaws, their spinal cords, and so on. And you amputate anywhere here, and this thing will grow exactly what's needed, no more, no less, to give you a perfect limb. And that's when they stop. So that's the most amazing thing about regeneration, is that it knows when to stop. When does it stop? It stops when the correct salamander arm has been completed. So this is some sort of means ends analysis, right? It's some sort of error minimization scheme, because the collective, no individual cell knows what a finger is or how many fingers you're supposed to have, but the collective absolutely does, because if you deviate it from that state, it will do its best to come back to where it needs to be, and then it stops. By the way, this isn't just for worms and amphibians. Humans can do this somewhat, and mammals can do it. So we regenerate our livers. Human children regenerate their fingertips, and antlers in deer, which are a large adult mammal, grow back every year at a rate of 1.5 centimeters of new bone. So this kind of ability is not just it's not something that mammals couldn't do. And I want to give you another example, because everything up until now has been about external perturbation, so injury, but the ability of regulative morphogenesis to overcome injury. But there's actually an even more interesting example. So this is a cross section of a kidney tubule in the newt, and this was discovered back in the 1940s. If you take a cross section, you see that the normal lumen is within about 8 to 10 cells that work together to form this kind of tubule. What you can do here is you can make sure that the early divisions end up with cells that have more genetic material than normal. So instead of 2N, you can have 4N, 5N, 6N, and so on. So more genetic material. The first amazing thing is that if you do that, you still get a perfectly normal newt. So you can have multiple copies of your genome. Apparently, that's fine. Then what you find is that the cells actually get bigger to accommodate that genetic material, but the newt stays the same size. And this means that fewer of these larger cells get together to form exactly the same lumen. So that's also pretty amazing. The cells adjust their number to their size. And then the most remarkable thing of all, if you make truly gigantic cells, one single cell will bend around itself, leaving a space in the middle to give you that same lumen. What's wild about that is that this is a different molecular mechanism. This is cytoskeletal bending. This is cell-to-cell communication. And so that means that it's an example of a kind of top-down causation. In the service of a large-scale anatomical goal, different molecular mechanisms are being called up to execute. And just think about what that means for a newt coming into the world. Evolution had to produce not just a solution to a problem of a newt environment, but actually a problem-solving machine, a second-order kind of machine, where you can't count on how much genetic material you're going to have, how many copies of each gene. You can't count on the size or the number of your cells. You can't count on not being separated by a scientist during embryonic development. Can't count on any of that. You have to be able to complete morphogenesis despite a wide range of not only external deviations, but changes in your own parts, right? I mean, we don't have anything remotely like this in our technology that can tolerate not only injury, but actually changes in its own composition and still get the job done. So this is what I mean by a kind of anatomical intelligence. It's a problem-solving capacity, which we still do not understand. And so the idea here is that this process is not simply complexity. It's not just feed-forward emergence. This idea that, and this is what's in all the textbooks, is that there are gene regulatory networks. They make some proteins, and then you get this process of emergence and complexity, where simple rules get activated, and eventually something complex happens. And here you go. You've got this structure. And of course, this all this does occur. Right? There are many ways to get complexity out of simple rules, but there's something much more interesting here, which is that it's not just complexity. It's the fact that if you deviate the system from this particular outcome, there are these loops that kick in both at the level of physics and genetics that will try to get you close back to where you were, navigate back to that region of morphospace, and in fact take different paths and do different things to get there. So it's not just a feed-forward kind of system. You might think about this as a kind of anatomical homeostasis. Now, on the one hand, biologists know all about feedback loops, and so that's obvious. But there's some stuff that's quite different here. First of all, Typical feedback loops have a scalar as a set point. It's temperature or hunger level or pH or something like that. But that's not going to work. In this case, you need some sort of descriptor. It's a shape descriptor. It has to have more information to it than that, even if it's not down to the single cell level. But also, we're really not encouraged, especially in molecular biology, we're really not encouraged to think about goals. We're not encouraged to think about processes that have an endpoint that they are trying to reach. That's considered sort of unwelcome anthropomorphic talk. And we're really just supposed to think about emergence and how the simple rules will give rise to whatever it is that they give rise to. But since the 40s, we've had cybernetics. So I think we can talk about systems with goals now. That isn't magic and it's not scary. And what this does is make some very strong predictions. It predicts that if something like this is true, and it has what every homeostatic system has to have, which is a set point encoded somewhere. If it has some kind of biophysical mechanism that encodes the set point, we should be able to do something interesting. We should be able to change the set point without rewiring the machine. So not make changes down here, which are actually extremely hard to know what to do, right? This process is not reversible. And so if you wanted to make a change up here, in general, we have no idea what to change down here. That's what's going to limit all the genetic editing, CRISPR, all that stuff is going to reach a ceiling after some low-hanging fruit of single gene diseases, because it's in general impossible to invert this. We have no idea what to change down here. But if we could change the set point, then maybe the system would just do what it does best and implement that set point. So this means that we should be able to find the encoding of the set point, we should be able to decode it, and we should be able to rewrite it. Okay, and that's what I'm going to show you now. But interestingly, so we start to think about, okay, how is it possible that cells and tissues store a memory, a pattern memory of what it is that they're trying to build? How could that possibly work? Well, we have to note actually that in order to have memories about large-scale states of affairs, you need a kind of cognitive glue that binds together the components. So we have an example of this from neuroscience. So here's an animal that learns to press the lever and get a delicious reward. Well, no individual cell has both experiences to form that associative memory. So the cells at the bottoms of the feet interact with the lever. The cells in the gut are going to get this the sugar that comes from it. No cell has had both experiences. So who owns this associative memory? In order to own that associative memory, you have to bind all of these cells together into one collective agent that has memories, goals, preferences, and so on, that none of the components do. And we know how this works. Well, we don't know how it works, but we know the architecture in neuroscience, and that's bioelectricity. So the thing that binds the neurons in your nervous system together to make up an organism that has goals and preferences in other spaces, what we have is some hardware. And these are ion channels that sit on the outside of these neurons, and they set a voltage gradient, and that voltage gradient may or may not propagate to their neighbors. And so now you have a network, you have an excitable medium, and it can do computation because information propagates in a very complex and regulatable way through that network. The software, it looks something like this. This is the physiology. So this group made this amazing video of a zebrafish brain active during as the fish thinks about whatever it is that fish normally think about. And so there's this project of neural decoding, this idea that if we were to read out all of this physiology and decode it, then we would have access to direct access to the memories, the preferences, the all of the kinds of cognitive content of this mind, right? That's the commitment of neuroscience, is that all of that stuff is literally in the physiology here. But the amazing thing is actually that every cell in your body does this. So all cells have ion channels. Most of them have these electrical connections to their neighbors. And this is actually evolutionarily where the tricks of the brain come from. It comes from this much older system that evolved around the time of bacterial biofilms, actually. And so we started to wonder, could we not port all of the things that are important about neuroscience, which is not the details of neurons, but the multi-scale cognitive scaling, to try to do a kind of decoding on the collective intelligence of the body? Could we read and interpret this information and see what it's thinking about? And could we read out these goal states?